All right, welcome everyone back to the Bioinformatics Lab podcast. I'm Kevin Libwit with Andrew Page. We're from Theogen, and today we're going to be talking about the use of next-gen sequencing in bioinformatics in the world of foodborne pathogen surveillance, something that both myself and Andrew have a lot of experience in. And Andrew, you've been working in this field for decades now at this point.
Oh God, don't say that now. You make me sound old. But yeah, so like I was working on foodborne pathogens somewhat in the Sanger Institute years ago, and then I was in the Quadram Institute, which used to be called the Institute for Food Research. So, for 115 years, it was always just doing food, and expanded out from there. So I have been doing food for a long time. And I'm currently on a UK government committee with the Food Standards Agency called the Advisory Committee for the Microbiological Safety of Food. It's a bit of a mouthful, but basically what they do is they do, I guess, risk assessment for foodborne threats, obviously with microbes, for the UK government. And it's an important thing. Mostly food in our world is actually really, really safe. But occasionally there can be very high consequence things that come along, and you need to deal with those, or you need to update things to deal with modern advances, like dark kitchens. That's a new thing. Like when you go onto Uber Eats or some kind of app and you order something, often it can come from a random place that you don't really know what it is. It could be in a warehouse, it could be someone is renting space in a cafe, in a cafe kitchen to do something at night. And so there's extra new threats that people had never considered, 30 years ago when they were making a lot of regulations. And the same with food delivery services. We have like HelloFresh and a load of others that deliver food to your door. But what are the threats there? Like meat being in cold packs or whatever, getting to your door. Is it okay? What if it takes a little bit longer? What if the truck is a bit too warm? This kind of stuff. So there are many new things that we have to consider, and so it's always good to keep on top of that before a problem occurs.
Yeah, that's fascinating, especially that business model of dark kitchens. I don't think I've ever heard that term, but as soon as you explained it, I knew exactly what you talked about because it's
What do you
very
call it in the
common.
US?
I don't even know. It's maybe I don't know the term. I was going to use the word phantom kitchen, but that's me just trying to figure
Ghost
out what to
kitchen.
call it.
I think
And that
ghost
makes
kitchens
sense
are.
because it'll be like an app. And what happens in the US too, it could front itself as like 18 different restaurants. So they kind of
Yeah.
like game the system on the Uber Eats, but it's all coming from one point source. So that could potentially be serving hundreds, if not thousands, around a locality. So yeah, the foodborne threats are that much more important to understand or rather addressing and surveying these foodborne threats need to keep up with these new business trends. And the transport thing is interesting too, because yeah,
Yeah.
the restaurant at that point has no longer any kind of control over the quality of that transport, the temperature, the proximity to other things. I don't know if somebody's got a pet in the back or whatever, level of cleanliness. You're depending on every aspect of from the kitchen to the consumer and what's happening along the way.
And then there's some very high risk foods, like, if you're the sushi, is it okay? We all know that the fish and whatever is raw and it's treated very carefully, and it's super high quality. But what if it takes an extra hour to get to you, after you've ordered it, or it's sitting around somewhere or whatever? You just don't know in terms of quality, and you can make yourself very sick if people aren't following the correct procedures. There's also another trend, which is people setting up, home businesses, and maybe they're cooking from their kitchen. I've seen leaflets come through my door here, for people selling chicken sandwiches, chicken burgers, and wings and things like that. But I know from the address that it's coming from a person's house, a home, and you're never going to have like the same level of cleanliness or operation procedures or separation of things, or even training, if you're preparing stuff in a home as you would in, say, a commercial kitchen. And, those are two very, very different things. And so, it is new levels of threats.
Yeah. Oh, and what you've kind of highlighted on is on the micro environments of the localities and those different kinds of kitchen environments. But then also continuously evolving is the macro scale. I think we've talked about it on the past before, the salad from all over the world. Oh, what is it? The well-traveled salad, right? So we have
Yeah.
super complex supply chains, getting food in from all over the world, plants or plants, agriculture, meat products, dairy products, all the different like. And then in the US too, there's the ongoing conversation of people's autonomy in eating different types of food, pasteurized or rather unpasteurized milk, as an example. Yeah, there's the growing
Oh,
trend and communities who, want to make sure that they have access to these things. So, food
and
safety.
unfortunately, there are also the people who don't like vaccines either, so
Yes.
something like diphtheria, is occasionally pops up, and that's a really old disease that we got rid of because of vaccination and
Yeah.
pasteurization. And if you're not doing either of those, like, my gosh,
Yeah,
crazy.
and we're speaking generally from the Western world of things, but then also in different parts of the world, especially where it's a little bit resource scarce, the understanding the supply chains, the cleanliness of food are all that much more amplified in the way in which it can spread, and there isn't necessarily that source tracking and shutting down and taking foods off the shelves and all the like and resources we have here in the U.S. or across Europe.
Yeah, I know in the UK, they went and did a survey of food packaging and,
Mm,
like I say, chicken in packets. And it was like 90% had Campylobacter jejuni on the packet. Like
mm,
that's a lost cause at that point. It's so ubiquitous. And
yeah.
clearly, it's clearly, I don't know, something spills on an entire pallet, gets contaminated very quickly. And, so people like in the UK think, oh, salmonella is the biggest threat from, say, chickens. But actually, I know that most chickens are vaccinated, say in the UK, and most they get imported. And so actually, that actual problem isn't really there. It's a lot of the disease people get is actually just Campylobacter, which it's usually not fatal for people who are young, fit, and healthy. But if you're immunocompromised or whatever, it can make you very, very poorly. And of course, then when you have the cumulative number of days lost in work and stuff, then it all adds up. So sometimes I think we can focus on the wrong threats.
Yeah. All right. So another aspect of it that we kind of highlighted the evolving and continuously dynamic variables we have to consider with respect to new business trends, complex supply chains, and all the like. But I think the world you and I are most familiar with is on the laboratory side, the evolving technologies that allow us enhanced capabilities to assess what's happening. So, for example, on my end, I came into public health amidst one of those transitions to the technologies that we know best. I came into public health, I think around 2015, which is during the transition in the US public health system from traditional molecular techniques like pulse field gel electrophoresis into the adoption of whole genome sequencing and bioinformatics as the main technology to support surveillance. And it was an interesting time because it was really the best time to be in public health as a bioinformatics scientist, in my opinion, in the US, because I got that front row seat to see how does a whole nation's public health system transform to implement these technologies that are clearly outperforming pulse field gel electrophoresis. And to some degree, we're still going through that transition, but there's been a ton of lessons learned over the past 8 years that we're seeing finally come to fruition and help us not only address the foodborne side of things, but also these other infectious diseases. And a lot of those technology implementation and best practices helped us even during the COVID-19 pandemic.
Absolutely. And you could see like some people who were working in food moved into the COVID response as well because there's similar skills are required. But what I really find fascinating is how recently, because they're doing so much sequencing, say in the US and the UK, of foodborne pathogens, you can start to link together so many different things and you can see multi-country outbreaks happening and you can stop them as well. It's that ultimate goal of spotting an outbreak quickly enough, stopping it, and then that's a good public health intervention. But I understand there's probably too many outbreaks to even investigate. So they have to pick the worst threats.
Yeah, it's a crazy conundrum though too. And I would say though, at least with whole genome sequencing, what we're also identifying is that it helps to prioritize when actual outbreaks are happening. I think that was the big revelation when you're looking at something like pulse field gel electrophoresis versus whole genome sequencing. You have the resolution of the proper outbreaks. You can actually um, get a higher level of granularity beyond just PFGE patterns. Like if you have the same PFGE pattern, that's as far as you can get in terms of relatedness. But as soon as you map over whole genome sequencing, be it through MLST schemes, whole genome MLST schemes, or even just using something like SNPs, you can break it out a little bit more granularly. You can see where the outbreaks are happening. So I think to a large extent, it also helped to identify how to best allocate maybe the epidemiological resources to investigate what's happening, as opposed to broadly chasing down all these potential leads from PFGE.
Yeah, what I found over the years is that every time people come to you with a new problem or a new data set or new study, and they have a particular hypothesis, usually it's wrong. As soon as you look at the genomic data, it's like, no, we can absolutely prove that, or we can absolutely rule these things in or out, and there's no ambiguity there. Your hypothesis is, it's based on old school,
Yeah.
epidemiology, but actually we know this is absolutely correct. It's because, in a lot of cases, when you go and do these reports and contact tracing, people maybe misremember, or in a lot of cases with foodborne pathogens, it's not what you ate, this morning or yesterday, it's what you ate maybe a
Yeah.
week ago. And people have bad memories. They don't necessarily remember what the original cause is. Some people might say, oh, well, I ate a bad batch of sausages, yesterday for dinner. And actually, it was the chicken you ate a week ago that was the problem. And it just takes time, to get into your system and then cause problems. There's very
Yeah,
few
that's
things,
the
giga toxins kind of stuff. Like there's very few things that are just like instant.
Absolutely. I think that's the difficult part on the epidemiology side. So, it is amazing to see on the lab side, on the bioinformatics side, how quickly the best practices have emerged. And it's not so much a question of, hey, which algorithm, how do I analyze this? It's more about the access to the bioinformatics tools. Because I think at this point with whole genome sequencing, and I think we've mentioned this maybe on a few episodes in the past as well, there's more or less, exactly what the laboratory is going to do in terms of analysis. They're going to do quality control to remove the poor read data. They're going to do a de novo assembly, and from that they're going to do the characterization in terms of identifying the relevant genes of virulence or antimicrobial resistance, and then they're going to build some level of phylogenetic tree through whole genome MLST or SNP approaches. So those best practices have given us that insight, and like you said, really indicate the true relatedness of different samples, and those have been tried and true, and there's countless publications showing the utility of these exact methodologies.
Absolutely, yeah. And I think for the future, we're gonna move to even better methods. Like for example, direct from primary samples rather than having to culture. That seems to be the trend at the moment when people are certainly with clinical
Yeah.
isolates is not to culture because it's expensive and takes time. Yeah, so it's quite interesting, that people are moving towards PCR-based approaches for detecting pathogens, and it gives us less samples, unfortunately, to do whole genome sequencing on. We can't do the same phylogenies, but actually, it's cheaper and better, and so maybe we're all good.
It's funny because as soon as I was talking about having those established best practices in the analytics, this whole conversation of CIDTs, and again, we've mentioned in the past, disrupts that completely. We won't be able to have the whole genome sequencing. We won't be able to do that tried pipeline anymore because we won't have those tools to necessarily just do QC, de novo assembly, and phylogenetic tree building. The characterization even alone is going to be quite difficult to associate certain genes with the actual pathogen that's causing the disease, and then the phylogenetics on top of that to have enough confidence in the metagenomic assemblies to perform the relatedness assessment is a bit blurry right now.
Yeah, I know as an intermediate step, say standard biotools used to be called Foodime, are doing these multiplexing systems. So basically amplifying up, say, each gene in Salmonella as an example.
Yes.
And then it allows you to do like a CGMLST. And so from a primary sample. So, you can very rapidly get most of the information. You don't get it all, but you get the most useful information for, say, CGMLST. So you can do typing, sequence typing, and which is quite useful. And of course, from primary sample, it works really well. So, it saves you that step. And so rather than just doing a straight PCR.
Oh, and we're seeing that, yeah, looked at pretty closely by folks in the US as the supplement at this point and potential replacement for whole genome sequencing. And so I haven't seen a ton of the bioinformatics downstream workflows. I've heard a lot about the wet lab techniques. I'm not aware of, or maybe it just hasn't come across because we haven't seen that data quite yet of what the performance is looking like. I don't know if you've gotten a chance to see, okay, how does this relate to if we had the isolate assembly and map to an allele database versus strictly the amplicon data targeted across.
I mean, last I heard, they're more focused on making the wet lab stuff work. I'm sure, everyone forgets about bioinformatics and how difficult it
Yes.
is. So I'm sure that'll be tacked on the end. And in a rush rather than being designed in from the
Right.
first place. And it is important, and like it's very frustrating actually when you see these huge projects and they focus so much on, let's say, one area, say on wet lab and protocol development, and then the bioinformatics is kind of this little sliver they hope will get done, magically by the magic button, when actually you do need, very high quality and highly skilled data scientists and bioinformaticians to actually go and analyze the data and pick it apart and really identify things. And they should be at the very early stages of experimental design, working all the way through, because everything impacts everything else, and you don't want to tack it on the end.
And you need scrutiny across these bioinformatics pipelines and processes as well along the way, because I think there's going to be a lot of nuance to this, because ultimately it needs to be super robust and relatively, packaged for the distribution at the scale that, the intent is to be, to replace everyone's whole genome sequencing and bioinformatics in the bionumerics of the world that we live in. It needs to be pretty solid and sound to be able to support international foodborne surveillance here.
Absolutely. Anyway, that's been a great little discussion. So
Absolutely.
I talk more next week, and thank you very much for joining me on the Bioinformatics Lab podcast.
All right. Yeah, we'll see everyone in the next one.